Hello guys, welcome back to Learning with Isha. We're talking about the applications of biotechnology, but today's application is way more than just an application. It is something that we all have heard in our day-to-day lives. The word insulin, right? We all know that people carry insulin with them, right? And if people forget their insulins, how much they panic, okay? Definitely this must be a life-saving thing. That is why so much chaos around it. What is insulin, guys? Insulin is almost instantly related to the word diabetes, okay? What is diabetes? Diabetes, हमने सुनते हैं ना लोगों का sugar level बढ़ गया है, right? What happens in diabetes is the glucose level, the sugar level in your blood goes up, okay? Naturally we are taking insulin, तो insulin क्या कर रहा होगा? इस sugar level को कम कर रहा होगा, right? So today we are going to talk not just about insulin but how we can use biotechnology to get more of insulin, right? Because our demands are going up and we need more and more amount of insulin, right? So for that we are going to use biotechnology. So let's first try to understand what is insulin, okay? Insulin is a peptide hormone. Peptide का क्या मतलब होता है, right? Peptide means it's a protein hormone. ओके इसके बेसिक यूनिट्स क्या होंगे? अमाइनो एसिड्स. जब हम इंसुलिन का स्ट्रक्चर आगे स्टडी करेंगे तो हम रियलाइज करेंगे कि एक मैच्योर इंसुलिन में ए एंड बी चेन्स होते हैं. इन दोनों को मिला के 51 अमाइनो एसिड्स का ये इंसुलिन की मॉलिक्यूल होती है. ठीक है, ये हम आगे उसके स्ट्रक्चर में स्टडी करेंगे. फॉर नाउ इट इज अ पेप्टाइड एंड इट इज अ हार्मोन, राइट? व्हाट मेक्स इंसुलिन? It is synthesized by the beta cells, okay? By the beta cells of the pancreatic islets. मतलब, it is made in the pancreas. There are cells called islets of Langerhans present in the pancreas, okay? They are making this peptide hormone called insulin. Now the important question is, what does insulin do in our body? Like I have hinted before, insulin is an anabolic hormone. See guys, it is very important to understand in the first place कि anabolism और catabolism क्या होता है. Okay? So these are two words that are opposite in meaning. Okay? Anabolism means where glucose will be changed into a storage form. Let's say it will be changed into fat. It will be changed into glycogen. Okay? And catabolism naturally will be the opposite of it. So the stored glycogen, let's say you start running. Okay, you need more energy than your regular circulating levels of glucose can provide. Naturally, where will I get this more energy from, right? I need more glucose. So there will be breakdown of reserve materials. That is called catabolism, right? So insulin is what? It is an anabolic hormone. Anabolism में क्या हो रहा था? बन रहा था, storage, okay? So anabolic means it regulates the metabolism of fats, carbohydrates and protein. Regulates the metabolism in what way? Take a second and think the answer to this question. क्या ये इनकी synthesis को promote करेगा या इनकी breakdown को promote करेगा? Like I told you, anabolism means it is going to promote the synthesis, right? So whatever glucose is circulating in my blood, it is going to promote the uptake of this glucose and conversion into fats, carbohydrates and proteins, okay? So what happens at the tissue level is that there are two things. In the tissue level, the glucose is going to cause glycogenesis and lipogenesis. Okay? First of all guys, to understand the meaning of any word, split it down into more simpler words, okay? So if I break down glyco and genesis. Genesis in English, what does genesis mean? Let's go to the genesis of this. Let's go to the origin of this. Let's find out where my story started, right? This means genesis. किसी चीज की शुरुआत कैसे हुई, इसको क्या कहते हैं? इसका genesis पता करो, ठीक है? तो genesis का मतलब होता है formation. Where my story began, okay? Glyco, like I said, take a hint from my previous explanation, glycogen. So the formation of glycogen is going to be promoted. Now think, lipo kahan aata hai? Okay, lipo means fat. So it's also going to promote the formation of fat at the tissue level. Okay, now what is it going to stop? It's going to stop these three processes. Lipolysis, like I told you, lipo means fat. Lysis means breakdown, right? So अगर ये यहाँ पे फैट को बना रहा है तो यहाँ पे of course फैट को टूटने से रोकेगा, right? So it's going to prevent this lysis. Proteo means proteins. Proteins are being formed, so it's going to stop the proteins in the body from breaking down, right? I don't need energy. I need my glucose to be converted into storage materials, okay? And what does gluconeogenesis mean? See, neonatal cardiology. क्या मतलब होता है इसका? That study related to the heart that is related to neonates, newborn babies. So neo means new. Gluco means glucose, new and synthesis. So इनको साथ लाओ, glucose new synthesis. So it is the synthesis of glucose from other compounds like pyruvate etc. Okay. So इसको भी glucose को तो हमें absorb करना है ना, तो synthesize क्यों करेंगे? So insulin ये functions perform करता है हमारी body में. Okay. Now in the next part what we are going to look at is कि ये insulin पहले कहाँ से मिलता था और फिर हमें क्यों उसमें क्या problems थी कि हमें biotechnology तक जाना पड़ा. Okay. Now we start talking about why do we need insulin from outside the body, right? There must be some defect going on in the body. What happens is when my body is not able to keep up with the rising level of glucose, right? I don't have sufficient insulin in my body. The glucose level will shoot up, okay? Beyond a point, it can damage my brain. It can damage my organs, right? So I need to supplement insulin from outside so that my glucose level will come down, okay? पहले कहाँ से ला रहे थे उसको? Initially, there were two scientists by the name of Banting and Best. Okay? Banting and Best were two scientists who first thought of doing this. Okay? So in the year 1921, they purified insulin from the pancreas of a dog. So if you get a question, if somebody asks you, where was insulin first purified from? The most common answer people give is cattle. But actually it was purified first from dog but later used in humans from where? From cattle and pig. Okay. So it was purified from here, but what was administered to humans was the insulin from the pancreas of cattle and pig. Okay. So what was the disadvantage of doing it? There were several disadvantages to it. Okay. Like I told you, insulin is a peptide hormone. Kindly try to recall how many amino acids were present in insulin. 51. I hope you guessed it right. Insulin is made up of 51 amino acids. So the pig insulin, the one that we were getting from the pancreas of the pig, differed from the human insulin at 1 amino acid position. In the case of cattle, 3 amino acids were different. Whenever we administer anything from outside our body, a word is used foreign. Foreigners होते हैं, क्या मतलब? जो हमसे अलग होते हैं, जो किसी बाहर की कंट्री को बिलोंग करते हैं. Similarly, any foreign molecule, any foreign biomolecule is what something that is not synthesized by my own body. It is coming from the outside. Okay. So the cattle and the pig ka insulin is coming from the outside. It does not belong to a human species also. There is a species variation also. So there is a high chance that my body is going to recognize it as a foreign molecule and attack it. I may develop an allergy. Okay. An allergy was seen in many cases. The second disadvantage was that if I have to get large quantities of insulin, just imagine the number of cattle, just imagine the number of pigs that would have to be killed, that would have to be slaughtered to get that, right? It's not an easy process. Also, it is not ethically right. Is it right to save one life and killing so many other animals? Ethical reasons also exist. Besides that, there is a risk of contamination. Okay, if the purification step, I took the insulin from the pancreas of the cattle. I tried to purify it. But what if it was contaminated with some bacteria in the handling procedure, in the isolation procedure, in the injection procedure? There are so many scopes for contamination. So, due to all these risks, what we decided? We decided to use biotechnology to synthesize insulin in the lab, and after that, those purified samples, those ultra purified samples were injected into the patients. So the next video is not only going to talk about this, but also going to talk about the structure of insulin in detail.